us. All right, Anne. Anne is currently the assistant professor of art and painting, the area coordinator at Metropolitan State University, Denver, and her practice combines digital sensing technology such as biodata sonification and analog traditional processes, including painting with ink she makes from locally sourced plant matter. Born and raised in Wilmington, Delaware, she earned her MFA at the University of Montana in 2019, and she was awarded a Fulbright Fellowship at the Natural Resources Institute, Finland, where she worked with restorationists to make collaborative art science work about former peat extraction sites outside Oulu. Am I pronouncing that correctly? Okay. But I'll let Anne tell you a lot more about what she's working on and a lot more about her practice. So thank you so much, Anne.
Thank you all so much. I'm going to talk to you a little bit about what led to the work that I have up in this gallery, and then a few other pieces that were more kind of direct collaborations with my groups. And in the middle, I'll tell you about this thing. So this is where I grew up, and I think this is subconsciously what got me thinking about like changes in the natural environment and how they shape humans. So we're at this 200-foot drop-off along these big cities. And that's like the point at which ships couldn't navigate the river anymore, and you could build a mill, and that just completely shaped our settlement patterns. We didn't feel like we had a lot to do, so we would make our own fun over the weekends. And looking back, I feel like this is sort of a beginning of a studio practice for me, engaged with natural elements. It was at times very frustrating or elating. I can't remember which in these photos, but that's me having a big reaction to it. But I thought I was a painter for a really long time, until I got to Missoula, and I saw this view around the back of Mount Sentinel. And in Delaware, you can never have a grassland like this unless you cut the trees down. And in fact, two sides of this mountain looked this way. So I was very lucky to meet a plant physiology researcher who was basically starving ponderosa pine seedlings of water, who was able to explain how this worked. And this is his slide. So this tree that seems static is under like atmospheres of pressure, pulling water up against the force of gravity. And the water is sort of snaking its way up these cells. And if one cell embolizes from drought, it can reroute, but if too many embolize, the plant will die. And this was really reminding me of some mechanisms that keep us alive or cause us to die. And also thinking about this idea of tension and how can I kind of show that. So I made work for a few years about ponderosa pines. And part of that work was using this galvanometer sensor. So you can attach these EEG pads to leaves of a plant and hear changes in what's happening in the surface of this plant. But the thing that I love about it is you can't actually hear anything truly from the plant. You're only hearing this sort of human assigned musical kind of filter of what we have decided we're going to do to sonify this thing. So it's data, but it's entirely subjective at the same time. And this got me thinking, still thinking about this idea of experiencing data or performing it instead of like collecting it and analyzing it, just being with it and letting it go. Okay, so that brings me to the pieces that are out in the gallery. Two quilts about peatland in Finland. So I was very lucky to get to go because of Fulbright. And this is my host organization. Luke is its name for Luonnonvara Keskus. And then this is bringing a whole studio on a plane. Very, very challenging. And here's where I was. About a 3-hour train ride south of the Arctic Circle in Oulu. And just to set the scene for you, here are some things that Finnish people could do. These ones in the summer, like, they all have a summer cottage and kind of harvest some mushrooms and blueberries. And then in the winter, there's this floating sauna boat in our town in the river. when you heat up enough in the sauna, you need to take a break. And so you do this. And it surprisingly feels really amazing. But I came there to look at this plant that survives this really extreme Arctic environment, and that is sphagnum moss. And I'm holding a long dangly piece of it here, and this is one of the conserved peatlands near where I lived. And if you zoom in on the plant, they're really interesting things. So this is the capitulum, the top of it, the only part that's alive. And then there's also this sort of long, snaky, entangled, enmeshed stem that starts hummifying, so it's not totally decaying, but it's not alive anymore. The plant is all tangled up with itself. It can make an artificial water table, which means if you're a small tree trying to grow on a peatland, once you get heavy enough, you will drown yourself. And if you zoom in even further, there are more cool things. So this is another microscope photo from the peatland that you saw earlier. These are the hyaline cells, and they're sponges. They can absorb 20 times their weight in water. And this means the peatland has engineered for itself this waterlogged, in this case, acidic environment. And so because there's no oxygen, nothing can break down, micronutrients don't become available for pretty much anything else. And so this really felt like it reminded me of us. This plant is making this environment where mostly only it can really survive and thrive. But of course we can find a way to use it. And so in Finland, being energy independent from its next door neighbors is really important. And so they will drain and kind of scrape away the peatland soil from these sites, and you can see what that looks like. They'll take a truck of this every 10 minutes to this power plant in the wintertime. In the summer, they don't even turn the power plant on. And so it's a pretty efficient kind of use of this fuel, and it's a biofuel, but it's not renewable because it's this really sensitive plant and ecosystem. And so there's been no way discovered to regrow this sphagnum moss once it's been taken away. And we had a day where we got to fly a drone at work, and you could really see the kind of scarring of the land from removing all of this plant matter. And you can also see the squares where things are growing are the study sites of my Finnish colleagues, and they have been testing different ways of amending the soil for about 10 years to get to that point. So I started making some paintings to try to, someone else has some input on it, to try to get to get to know this plant and the ecosystem a little more. And I also was really interested in using materials from the site. So this is the ash that is industrially produced from burning the peat. So we have a lot of it. So I made some paint out of that. Out of fungi that grows there, spruce, some other things, some of the paintings, my desk full of plants. These paintings were shown in this biennial in the town of E, spelled with 2 I's, called Knowledge of the Earth. But I got to use another really cool tool at this science institution called the hyperspectral camera. And it looks sort of like a regular camera, but instead of this 2D set of pixels, for every visible pixel, we also get this wavelength or this set of wavelengths of information that we can't see. So they call it a data cube. And this is kind of what it's telling you. So all of the stuff we can see is kind of crammed over here, and then there's all of this data about water content and cell health, like all of this interesting stuff that we can't see. Here's one of our soil core samples on the camera bed, and here's what it looks like in the analysis software, which was weirdly partly in English and partly in Finnish. It was an unusual challenge. So we have a false color image here, and then when you click on one pixel, and it's hard to see, but one pixel is put there, you get this view. And so I started thinking, we can see what's happening with these wavelengths of soil data when we hear them. And so I've mapped this out onto staff paper. And so we have a fertilized core sample on the top staff. So the peat ash has been added back in. They think that's the best amendment. And then the control sample and the dynamics and the tempo and everything are controlled by different parts of the soil data. And then there are also these breath marks where there was no reflectance in the hyperspectral data. So we think like where there's air in the soil. So I started thinking like, could we have like some sort of breathing thing happen to show this aeration in the soil? And I brought these so I can pass it around if you want, or I could leave them here for you to look at later. Like, now. Okay, amazing. There's more. So I sent this to a choral director in Finland, and he said, we can't deal with this weirdness. Come back when you have something that looks more like normal music. I was like, okay, sounds good. And I sent this to my friend who's an electronic composer, and we've worked together, and he's kind of used to this strange thing that I do. This piece went in this collection. I'll come back to the sound in a minute because we're getting close to how this quilt works that's outside. So I still had all these plants on my desk, and I decided that papermaking is a big industry in Oulu, this town, and so I would do that. And so they put the artist in the soil sample room in the lab, so we're like all the messy stuff together. And this is after 4 hours of boiling with washing soda. And then this really technical part of the process was just to stomp the fibers on the parking lot for the length of about 1 album. So very sophisticated things you can pack that in a suitcase. And here is kind of the forming of the paper to the sheets. I made them into a grid and projected this map of the extraction site onto them, and then began embroidering these tracts of land that we took away, filling those back in. And the hand embroidery started to feel to me like this analog of the labor of the peat moss, as it grows so slowly with less than a millimeter in here. Then I tried attaching transducers to it, ended up attaching speakers, and then through those speakers and this pedal system, this is what you can hear. You have to be a little patient, and it has to be quiet in here, because the first part of it is not like really the audible range, but you can hear this electronic take on that graphic score of 160 years of soil data through the materials from the site as it is now. But I really still wanted to have this human voicing of all of these beings in the soil. So I got an alumni grant to go back to Finland in 2022, and I worked with a choral composer. This is her desk, and she's starting to translate this thing into something more conventional music notation. So I can pass this around too. That's half of them. So this is one image. So she kept some of this graphic notation, and she worked with the choir to find a finished poem about peatlands to be the lyrics. This is the Tawira Chamber Choir. It's 50 people. They've done some experimental stuff in the forest before. Here's their rehearsal that I got to go to. And here is where they performed it. New Music October. That's another finish lesson for today. And we also got to do a projection to go with the sonic performance. So I worked with a programmer, Brian Gibbons, and he was using processing visual coding language. And we came up with this plan together. So this is this image that has the data that we can't see. And this image, he programmed to rearrange the pixels in this image that has the plant that we can see. And I have, this is just one of many pages that he wrote, and I have that too, if you're interested. It's actually weirdly understandable, but maybe not in 30 seconds. And so what happens is we start with all of the pixels arranged based on this hidden image, and this was reminding me of the ability of peatland to also store a pollen record, because nothing's breaking down, so it's like this geologic record. And then gradually it sort of seems to assemble itself. So it sort of looks like that halfway through. Also on the trip I got to go back, that's my research host who's done all the science. Here's what's growing there now, quite a few more things. And this is like the seed of the next project. So I have a CO2 sensor and I'm measuring sort of the respiration of the ground. Don't know what I'll do with it yet, but I'll keep you posted. Also in Finland, this is the balloon piece. We got to do a residency for a week in Kilpisjärvi, which is a biostation kind of in the far northwest corner of Finland. So they say she's like a dancing woman, and Russia took her one arm. So the arm that's left, the top of that is where we were. And here was our sauna. So we would go here at the end of each day and then swim in the lake. And one time I was swimming in the lake at night and I saw this. That's not the picture I took because I didn't have my phone. It was in the lake. But it was really amazing. And there were several teams here to look at this theme of the heavens. And our team that HAV stands for High Altitude Bioprospecting. So we were really thinking about like who's living in the atmosphere over the Arctic? How did they get there? And also this idea of bioprospecting in the field of biology, very normal, kind of neutral term, but we're thinking like, what are the undertones of this? Like, are we using them or are we able to like, really communicate with them and collaborate with them? Right, our questions, our table of many things we brought from our separate countries, helium that we finagled up a trail to fill up our helicite, or a helium-filled kite balloon. Looks like this. And we ran a bunch of flights, flying a kite for research, highly recommend. Here's what it looks like in the air. It's about 200 meters high. We weren't allowed to send up a weather balloon because we were too close to international airspace. Like, it was very, on many borders, many man-made borders. Till is a sound artist, so he attached some microphones to this tether line. I was interested in like the kite make a drawing, and so we attached these mark making tools to that tether point also. Here's the science part. We sent up these antimicrobial copper wingstops, wearing the 3M suits just before COVID. There's a bunch of, it's just like a beautiful experience, deflating it, back in the lab. Here's what we started growing. Mel took these back to England with her. Was curious, like, will I be allowed to go through the airport with all of these beings? Did PCR, and we were able to identify a bunch of them. And then now we're all in lockdown, and we're back in our separate places, on a bunch of video calls, drawing these things, trying to understand them. This one was first isolated in Antarctica. So we're wondering, did it travel this whole way, or is it sort of ubiquitous in the atmosphere? They started to look really floral to me, and I was thinking about this still life era when we were kind of naive about microbes, but we had this sort of hints of death and decay, this fear of them. So we were all drawing. Heidi, our collaborator in Finland, digitally stitched these back together, and then they were woven on a jacquard loom into this piece, hengus. And hengus is the Finnish word for fabric and also forest type. So I was really interested in like, why are those two things joined together? This idea of enmeshment. Again, here's a closer view of this sort of grid structure, and there's this really awesome rhizomal thing happening on the back. And we were really interested in, like, so much of what we were doing on site was very analog, like this old-fashioned kite flying thing, and then getting the data was this really sophisticated digital process. Like, what do we gain and lose in each of these? And here's some other pieces. We had a show in Birmingham, UK, and we moved to Finland. And then our leaving to Italy, I was like, this piece is traveling, like, I want to go with it. And then we have a soil dialogues project. So this is a group of about 30 artists from around the world, part of Eco Art Space. Tali is part of Eco Art Space too, that's how we met. And we've been meeting online since 2020, thinking about, could we make a piece collaboratively from our own homes to do with our own soils and then somehow stitch them together, quilt them together. And we did that for this conference in Florence this past summer. And I was working at the time with soil in Pontotoc County, Oklahoma, right there in the Cross Timbers region. So I was really interested in this. This is what it looks like from above ground, how I usually was exploring it. This intersection of eastern and western species and drier and wetter climates. And so what we all did in our separate places was bury cotton fabric underground for a month or 2 months, and come back and exhume it. And the microbes have made the first marks on the piece, or sometimes completely kind of eaten it. And this is what I came away with. It's like kind of like a tie-dye with beings in the earth. And a first layer of a painting to react to. And so we also sent a small soil sample to a lab in Texas, and they provided us with the hundreds of bacteria and fungi that were in this like tiny fraction of the Ziploc bag. And so I made these kind of characters based on the most common ones, and started painting those in response to the marks they theoretically had made. I animated those paintings together, and then I started working with collaborator Sherry Feldman, who's this experimental vocalist. Started thinking, like, can this painting animation be read as music notation? This is the data we got. There's pages of this. This is what it looks like when it's all rolled out. This was at a conference. And I started thinking, like, okay, these are the categories. Don't feel like you have to read this. But like gram-positive bacteria has this like thicker wall. So can the sound have this sort of thick edge to it? Or we have different shapes of bacteria. So the spherical ones, can they have this sort of round sound? Or the raws, the bacillus type, can they have this woo, 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 like elongated thing? So I gave a bunch of pages like this to Sherry, thinking like, this is the warty microbe, the verruco microbiome. So can the sound have these sort of like distorted growths to it? So I handed her, I think, like 40 of these, and now I'm going to hand them to you in case you're curious. This was the text from the exhibition. And here's one of the pieces, the collaboration between me and the Oklahoma soil microbes. Here's where they were shown. This was the palazzo where the conference was. We really could do better, I think, in America. And this is the view from where we were talking, which was very distracting and amazing. And then we had a pop-up exhibit as part of the soil conference. We were kind of infiltrating all the science with a bunch of artists and anthropologists. So we were at an old convent, which is now a cafe and a pizza restaurant, and also a set of art studios. And this was our exhibit, brought in suitcases. And here's what it looked like. So we had a little screening room, and a bunch of work made with this buried fabric technique, which I highly recommend. Easy to do, really interesting. And I'm gonna try to play it for you and put the microphone by the computer. It's very short. I don't know if that's audible at all. So I was really interested in like the layering of all of these sounds and thinking about like all of these microbes are coexisting in this tiny bit of soil, and that seemed like somehow an analog for this meeting of ecosystems in that Oklahoma zone. And maybe something about us as humans needing to live with people who are different than we are. So I'll stop it there. It's online, and you can see more if you want. And thank you so much for coming, and I'm happy to answer any questions you have. Any questions? It always takes me a long time to come up with questions.
So the sound that we hear with this piece out here, where did that come from? Was it created by a computer, or were you?
It was created by a person, but with like digital sound equipment. based on the score that I passed around that has wavy red, green, and blue lines. There is a little bit of field recording in there. It's audible at certain times and it's quiet of me walking on the peatlands, and it's this very like pillowy, soft, frozen, crunchy sound.
Is the plant life at all emitting some kind of sounds that we can't hear with human ear?
I think so. There's a researcher named Kim Rittner who was able to, I think with a contact microphone, actually record the embolisms of a plant in drought. And it just sounds like this set of like, and then I had a student, I'm teaching a bioart class in Denver, and there's a student who found some research on the sound of microbes themselves. They put the microbes on kind of like a drum head, and because they're moving and doing processes, the drum head will kind of amplify those.
When I was a little kid, back in the 60s, I was watching some TV show. This guy comes out and he says, I'm gonna prove that plants scream.
Yeah.
And he had this plant in a pot, and he had something hooked up to one of the leaves. And, you know, the meter reading the sound source was jumping a little bit. And then he pruned it. And when he cut it, that needle, like that, everybody in the audience screamed.
Yeah.
I didn't know if that really proved plants scream or that was just
I don't know, or like we like to assign a human emotion to something. That's really interesting. Yeah, actually in bioart class I get to.
Urgency to learn more about it recently or
I think so. The question, maybe you'll hear it, was like, do I teach about this work to students in Denver? Have they changed or has there been more urgency about it? And I think the bioart field seems like pretty happening in Europe and Australia and like maybe like starting to happen here. But there's a pretty amazing contingent in the Front Range area. And I have a colleague at my university who does it too. So I feel like there's some momentum there. And the biology faculty person I'm working with has been kind of interested in this and had students doing it kind of informally for a while. So I think so. And I feel like our students always have some urgency about their work. But their work seems to be a lot about identity, so I'm kind of interested in like, are we thinking about microbial identities in terms of our human identities, and like, are the microbes like representative of a marginalized population that we're not hearing from or like, not considering their rights or agency? Is that an easier way to start thinking about humans that we aren't considering enough, or vice versa? We'll see. But they make some exciting work. And I get to actually am working with some PhD students on this bison project. I don't know if I told anyone about the bison project. So we have a herd of bison managed by a Denver zoo just west of town. And 2 of these bison died and their carcasses were left on site. And my colleague is a soil chemist and she's been curious and measuring like what's happening to the soil nutrient levels kind of in concentric circles around these carcasses. And so for the first time, I'm gonna try to actually collaborate with students, not like I've given them like the same sort of prompts and restrictions that I've given myself, but we're gonna like make something together, kind of like this cross timbers Oklahoma project. And then the playground ensemble is this experimental music group, so they're going to use that as a score. There's a data soundification class that's happening in the music department to work on this. And then we're hoping that we can figure out how to animate it for the planetarium dome, so we can feel like we're underground at the site of the carcass. But we'll see. Always trying to do something new and difficult. Questions?
I'm curious if you've read any of Ms. Burrow or what you wrote the essay on thinking or copy materials. And I don't know about indigenous ecologies, looking at like in Durham or complicated allies, but the way their artwork and Durham and other indigenous artists sort of assign an agency and will to the materials that they work with, including stone, wood, grasses, and then thinking about the relationship between themselves and the material as stewardship rather than the human sort of acting on and with and nurture matter. And the way that you are sort of more call and response to some of the processes, and I think there's a way that the call and response obviously is very personal collaboration, right? Is the natural matters that you interact with together. I was wondering if you encountered this theory of how to figure out how to express yourself.
I haven't heard of them. I'll have to get them from you. But my collaborators in Finland were really into talking about Jane Bennett and this idea of vibrant matter. Like at some level, it's not inert. And you are like, what are we going to think of ourselves later when we're doing this like nano reading here? Did graphic agree to this?
Yeah.
We should trade. Maybe it's pastry time. Thank you all so much.
Thank you.